Good morning, everyone. So in my last video, I talked about overview of fatty acid synthesis. Today, I'm going to talk about steps of fatty acid synthesis. But before talking about steps of fatty acid synthesis, let's do a quick recap of the overview. You know, fatty acid synthesis is also known as de novo lipogenesis or DNL. And you know by now very well that it's a well-fed state pathway. You know what's well-fed state pathway? Well-fed state pathway is also known as high insulin state. High insulin state is a well-fed state. Now, what else is required for fatty acid synthesis? As I told you in my overview, synthesis always requires ATP. So yes, you need a lot of insulin as well as ATP for fatty acid synthesis. Please understand it well, the difference between well-fed state, which is high insulin state, and as well as well-fed state, which is high insulin, high ATP state. So fatty acid synthesis requires both. Now let's see what are the other requirements of fatty acid. To know that, this is the general structure of fatty acid. So to synthesize fatty acid in your body, you need a source of carbon, which is acetyl-CoA, and you need a source of hydrogen, which is NADPH. So let's complete the other requirements of fatty acid. You need a lot of acetyl-CoA to synthesize fatty acid. But remember, fatty acid synthesis is a cytosolic process, and that we learned in the overview. So this acetyl-CoA must be available in the cytosol for fatty acid synthesis. And also, we know very well that in a well-fed state, you get acetyl-CoA in your body by burning carb. You have to burn carbohydrate to get acetyl-CoA for the synthesis of fatty acid. What else you require for fatty acid synthesis? NADPH. And if you know carbohydrate metabolism, you know that you get NADPH from HMP shunt. HMP shunt is also known as hexose monophosphate shunt or pentose phosphate pathway. This is the major source of NADPH in your body. Why you need NADPH? You need NADPH as a source of hydrogen for fatty acid synthesis. But there is also a minor source of NADPH in your body, and I will talk about this enzyme in the steps, and that is known as the malic enzyme. So now let's start the step of fatty acid synthesis. And as you know that you get the starting molecule for fatty acid synthesis is acetyl-CoA, and you get acetyl-CoA from burning carb. So let's see how do you get acetyl-CoA. So you burn carb or glucose, and through glycolysis, you know, you get pyruvate. Now pyruvate enters the mitochondria through a transporter, and pyruvate is in the mitochondrial matrix now. So let's see that pyruvate from the cytosol moves into the mitochondrial matrix. You know the fate of pyruvate in the mitochondrial matrix. Pyruvate forms acetyl-CoA, and acetyl-CoA then enters TCA cycle with oxaloacetate. This is TCA cycle, and I'm sure you must have learned TCA cycle with carbohydrate metabolism. And this cycle is essential for synthesis of ATP. And you know that this is an important pathway of glucose metabolism, by which you get a lot of ATP in your body, TCA cycle. But now, how is this connected to fatty acid synthesis? Now, please don't forget that fatty acid synthesis is not only a high insulin pathway, but a high insulin as well as high ATP pathway. So in high ATP, this is the step was catalyzed by isocitrate dehydrogenase. In a high ATP state, do you think that the dehydrogenases will be inhibited in a high ATP state? Because we know that fatty acid synthesis is not only a high insulin pathway, it also requires a lot of ATP. What happens because of this inhibition? The substrates or the intermediates above the block accumulate. Citrate is pushed out and is made available in the cytosol now. Now we will also talk about regulation, but in the end, so let me first complete the steps of fatty acid synthesis. Once citrate is pushed out in the cytosol, you know this is cytosol, and this is mitochondrial matrix. So once citrate is pushed in the cytosol, citrate splits back into acetyl-CoA and oxaloacetate. Please know that acetyl-CoA is now, which is the starting molecule for fatty acid synthesis, will now go into DNL. What are the enzymes required? Well, we will talk about the further steps in my next video, but also know that oxaloacetate joins pyruvate, and we will talk about that too in regulation. What is this step required for conversion of oxaloacetate to pyruvate? Because as I told you that NADPH major source is what? HMP shunt. But what's the minor source of NADPH? Minor source of NADPH is malic enzyme. So we will talk about these two in my next video with completing the steps of fatty acid synthesis. Now the key is one important regulation you should know, that yes, when it's a high insulin and a high ATP state, isocitrate dehydrogenase is inhibited and the intermediates above this block accumulates, and that's why citrate is pushed in the cytosol to turn on DNL. But remember that this is not enough to turn on DNL. You still want a slow turn on of this pathway in the forward flow, so that you can have more and more citrate in the cytosol to do fatty acid synthesis. Now, who regulates that? There comes the insulin PFK2 regulation. You know what is insulin PFK2 regulation? Very important for your body. You must know that insulin turns on PFK2 by what mechanism? Always and always dephosphorylation. Insulin always dephosphorylates the key enzyme by dephosphorylation, and that makes PFK2 what? Active. When PFK2 is active, its product increases in your cells, which is fructose 2,6-bisphosphate. The fructose 2,6-bisphosphate, which is increased inside your cell, will then turn on one of the enzyme of glycolysis known as PFK1. This, because being active or because being turned on, both glycolysis and TCA is turned on a little more. Where is this happening? Because as I told you, not only that fatty acid synthesis is a cytosolic process, and that is why you need acetyl-CoA in the cytosol. Fatty acid synthesis major site is the liver, minor is adipose tissue. So this regulation is actually happening in the liver, because in the liver there is major fatty acid synthesis. And why is this required? Because in a high insulin and high ATP, especially high ATP state, these pathways of oxidation, glycolysis and TCA will be slowed down in most of the tissue. But in liver, you don't want these pathways to be slowed down. Why? Because one of the intermediate of TCA cycle is actually essential for fatty acid synthesis, which is citrate. So to increase the forward flow or synthesis of the citrate, who comes in for rescue to overcome the ATP inhibition over isocitrate dehydrogenase? Insulin PFK2. Insulin PFK2, fructose 2,6-bisphosphate, turn on glycolysis and TCA for a little more time only in the liver, please remember. Otherwise, in other peripheral tissue, the inhibition of isocitrate dehydrogenase by ATP still continues. And in most of the peripheral tissue in a high ATP state, the glycolysis and TCA slow down, except in liver. This is turned on a little more by insulin PFK2 regulation. Now in my next video, I'll explain you how acetyl-CoA finally forms fatty acid. Because the first fatty acid to be formed is a 16-carbon fatty acid, which has no double bond, known as palmitic acid. And as you know, we not only have saturated fatty acid in your body, which is actually formed by this pathway, we have polyunsaturated fatty acid also in your body. So then I'm going to further talk about how acetyl-CoA goes into DNL with the help of two enzymes, acetyl-CoA carboxylase and fatty acid synthase complex. And I will also discuss the regulation. We will also talk about the significance of this step in the next video. Till then, please keep giving your feedback. Thank you.